Good afternoon, everyone. So I'm Pierre Oesterle from Umea University, and I will be talking about hydrothermal recycling of activated biochars loaded with emerging contaminants. So this project, I've been doing it with my collaborator, Christian Porter, and also our supervisors, Stina Jansson and André Masek. Christian Porter is the one kind of hosting this webinar, and we got a lot of I got a lot of help from him because he's working also with the UK BRC, so the Biochar Research Centre at the University of Edinburgh. So first of all, we just talk about this cyclic pollution. So whenever you are taking a pill, it could be antibiotics, drugs, it could be hormones, for example. Not everything is degraded in the water, in the body, and so maybe half of this compound is going to end up in the wastewater. After that, you have this wastewater goes in the wastewater treatment plant. One of the problem is that they are not able to degrade this pollution. So in the end, you get those hormones, those micropollutants at micro concentration. They end up again in our river system and then in our drinking water, which means that you have an accumulation of those compounds in people, but also in crops, for example, for agriculture, where we are using water. So all of that makes that it's a very bad loop, where you could have an increase in antibiotic resistance from bacteria, for example, or you could also have some effects due to the hormone consumption, the daily hormone consumption. So one of the ways to remove that would be to put a sort of tertiary treatment on the wastewater treatment plant. And one of the most common way of degrading or removing those pollutants before they end up in the drinking water is to use sort of filters. Those filters with combination of ozonation or different techniques, they make us able to remove those pollutants before they arrive in our drinking water. One type of filter that could be used is, for example, activated carbon or activated biochar. So, as I just said, carbon material are quite effective at removing emerging contaminants, and this is a sort of filtration, so it doesn't adsorption. And so when those contaminants are absorbed on the char or the carbon materials, they don't leave the surface. Two different materials are used currently. It's activated carbon or activated biochar. The most preferred one is activated carbon. However, it has a higher price, but it gives also a higher capacity. So it can absorb a lot of activated biochar, have a lower capacity, so they cannot absorb as many contaminants, but they have a lower price. So the idea is that when you use those carbon, what do you do afterwards? Do you discard them, and how do you discard them? So one of the idea is to, after sorbing all those contaminants on the surface of these chars, then you want to regenerate the material so you could reuse it in a kind of a circular loop. One of the problem is currently the thermal regeneration is quite preferred, and the problem is that when you are using activated carbon in wastewater treatment plants, the material is wet. So as thermal regeneration requires dry material, you need to dry it, and then you use a lot of energy, which is of course a lot of money spent to just dry the material, and then on top of that doing hot gas stream to just degrade the contaminants. So it's not very practical, and it's very unsuitable for biochar. And one of the main reasons is because the price of manufacturing biochar is quite low. So if you are using a very advanced technique to just regenerate it, then it costs more than just put it, for example, on a long-term carbon sequestration in the soil, or just burning it off rather than regenerating it. So the aim of this study is the development of another type of regeneration or recycling method. And this would be the way of degrading those emerging contaminants, but in a more efficient way. Okay. How do we do that? Well, we have been thinking with Christian that hydrothermal carbonization is a good way of doing that. So hydrothermal carbonization and hydrothermal recycling is basically you are using a pressure cooker. When you're at home, you want to get you want to soften faster your vegetables. What do? You use a pressure cooker. This is the same system that we're using to do carbonization of wet material, wet feedstocks. That could be, for example, sludge from paper mill industry. With that you can make biochar, or it's called hydrochar. In the same way, as we have already this biochar, if we put it in the same conditions at high temperature, high pressure, so it varies between 150 degrees to 370 degrees, with high pressure 0.4 to 22 megapascal, you are in solvolytic conversion conditions. And with that, you could kind of increase the efficiency of the degradation and do that quite fast. And as said, it will be quite cheap because we don't have to dry the material. We just have to heat up the water. So our hypothesis was that biochar, they will remain stable in those conditions, because we have already been making those biochar or activated biochars at quite high temperature, which is around 550 to up to 800 degrees Celsius. So they're quite stable. However, the contaminants that are sold on those jars will be less stable, and that's what our guess was, our hypothesis. On top of that, we were thinking that we could enhance the degradation by just using catalysts. And so we were thinking about adding catalyst to the biochars and then do the same type of reaction and see the differences. As I said before, the advantages, of course, are that you don't handle the dry carbon, you have a lower energy consumption because you don't dry them, and you have a possibility for inline treatment, so you could just take and then degrade those contaminants and put the material back. So it's kind of very good in terms of logistic. Our experimental step was divided into 4 steps. The first one was the biochar production, so we did 5 different biochars. Then we solved those 10 different contaminants on those biochars. And then we did the hydrothermal treatment. And in the end, we extracted the contaminants that were still remaining on the char surface, but we also analyzed the liquid phase. And with that, we can determine if the method is good to degrade those 10 contaminants. Regarding to the biochar production, so we have been using 2 different sort of feedstock, the softwood pellets and wheat straw pellets. Those ones were provided by the UK Biochar Research Center, and they are quite good because they are standard biochar. So they have them in a very large amount, and then we could have reproducible results because we exclude the variability of the material. And we have chosen softwood and wheat straw because they also have differences in their compounds, like lignin content and cellulose, hemicellulose. On top of that, what we have been using also is to make a mix of biochar and minerals. And so this composite, for this composite, we use the catalyst, which is ochre, and it comes from mineral waste. So again, it's a good way of using a waste and have an added value to it. And this ochre has been used usually in catalysts as catalyzing the pyrolysis or catalyzing hydrothermal treatment or carbonization, so increasing the reaction rate. For the production conditions, we have been doing that in UK. in Edinburgh. And first we pyrolyzed the char at 550 degrees Celsius under nitrogen conditions with the machine that you see on the right. And then secondly, we activated the materials at 800 degrees Celsius under carbon dioxide for 60 minutes. These are a little bit the results for the yield and the surface specific area. I will talk about the surface specific area. You can see that when you add ochre, you see a lower surface area. It's probably because the ochre is also going into the pores, so it's blocking the pores. And you see the last material, which is activated carbon, which was kind of a reference for that. It has, of course, a higher surface area, and that I showed it at the beginning, that activated carbon has a higher surface area, but a higher price. For the emerging contaminants, then we have been using 10 different pharmaceutics that were found in a wastewater treatment plant effluents, activated sludge, or surface water. And those, it comes from studies that have been done at Umea University and around Sweden. So those compounds are found after our wastewater treatment plants. I just want you to pay attention on two different compounds, which are caffeine, that almost everyone is drinking every day, so you know what it is. And fluconazole is also an antifungal medication. Both of them are quite difficult to degrade usually, especially fluconazole, because even with ozonation or other tertiary treatments in the wastewater treatment plants, it's very difficult to degrade. So we thought that it would be a good platform to see if our technique could work on very difficult compounds. Regarding the experimental conditions, we mixed those 10 pharmaceutics at 1 to 2 milligrams per liter, which is quite a high concentration. Usually in the nature or in the effluents, you get it in micrograms per liter or nanograms per liter. The idea was we wanted to solve quite a high concentration of those char, because when you are doing the filtration, then they get loaded over time. So maybe it would load up to 5-10 milligrams per biochars. What we did is that we needed to put that in deionized water, but also added some methanol, because some of those fatty compounds were very difficult to solve. Then we have been for the extraction. So after the experiments, as we were doing the extraction, I've been developing an extraction method that is using dichloromethane, methanol, and formic acid, and that works for those 10 contaminants at very good recovery. So this paper is in preparation for publication. And then we did the triplicate for each sample to just be sure that we have a representative sample and it's not a random thing. For the hydrothermal reactor setup, what we use then is we didn't want to have usual liners that we use in hydrothermal carbonization reactor, because those liners, if you reuse them, then you could contaminate those liners, and then you are not sure of the results. So instead, the reaction vessel that we use rather than the common liners was to use borosilicate glass tubes. It's easy, they are easy to use, and they are quite cheap, so you could discard them afterwards. And with that, we avoid cross-contamination. So we take the glass tube, we add the biochar sample, so 50 milligrams, and then we add a stock solution at 7 milliliter. To avoid any reaction with the air, then we fill it with nitrogen gas. And then we seal it with a torch. And there at the end, we have a sealed glass reactor. This sealed glass reactor will be shaken for 24 hours, so we are sure that almost 99% of the compounds are sorbed onto the char and they are not still in the liquid phase. From that, we put them in the hydrothermal reactor, so the pressure cooker, and we fill it with nitrogen gas, but also water. And the it's very important to add water, otherwise there's too much pressure that is building up on the glass head of the glass reactor, and then it explodes. So it counterpressures the pressure, and with that, we did that at 5 different temperatures, from 160 degrees to 320 degrees. So with that, we have a big span of temperatures to know which one is the best one, or which one is the most preferable one to degrade those contaminants. What about the results? Well, first I'm going to show you this graph. So it's the contaminants. So we first did a trial with only the liquid, the liquid solution. So we didn't use any char, and this liquid solution was containing those contaminants at 1 to 2 milligrams per liter. And then we did it at different temperature, and we wanted to see how well does just the hydrothermal reaction or hydrothermal treatment work without any char. As you can see, at 280 degrees, all the contaminants are degraded. So it's we were thinking, okay, it's great. 280 degrees is not too high. What about then the other biochar? So as an example, I'm going to take activated softwood pellet biochars, and we did the same. So I didn't precise, but on the left side, you have the remaining contaminants in percent compared to the initial concentration. So 100% means that you still have everything that is remaining in the on the surface of the char, and for the previous one, it was also in the water. And if you have 0%, that means that everything has been degraded. So here you can see that first I have to say that there has not been any pharmaceutical in the liquid phase. So that means that everything has been either degraded or has been still remaining on the surface of the char. So from that, we extracted what was on the surface of the char, and we could see a similar trend for all the samples. So this is kind of a representative of what happened. You can see that for fluconazole and caffeine, the degradation was much worse, or it didn't degrade very well compared to the other compounds, which means that we needed to have higher temperatures than 240 degrees to degrade most of the compounds. Now, if we go specifically for fluconazole, so in this graph you have the different type of char. So SWP, SWP5, which is softwood pellet at 5% with ochre, SWP10, which is 10%, and then WSP, which is with straw, and activated carbon, which was the reference, and just the liquid phase reaction, which didn't have any char when we did the experiments, is called no char. As you can see, the degradation is faster when you have biochar at 160 degrees compared to what is happening at 160 degrees Celsius when there is no char. So that's kind of promising. And then when you have the ochre addition, then you can see that the ochre for the moment at 160 degrees doesn't catalyze any degradation, and it kind of even leaves more contaminants on the surface of the char compared to without ochre. The opposite trend can be seen at temperature higher than 240 degrees. So as you can see on the right side, when you don't have any char, at 280 degrees, everything is degraded. However, on the other samples, such as softwood pellets, for example, there's still 35% of the contaminants that are left on the surface of the char. So we were thinking that this phenomenon was quite strange, but could also maybe explain that the reaction cannot penetrate fully into the char. So the char seems to hinder or protect the adsorbed contaminants at higher temperature. For the okra, we can see that actually we got quite good results when we increased the percent of okra. We could see that actually there was maybe a catalysis of the reaction, because at 280 degrees we see a massive drop of 35% of the remaining contaminants, and then at 280 degrees everything is then degraded, which is almost the same condition that we got it from when we only have the liquid phase, so no char. For caffeine, then that's the same trend, and that's actually exactly what has been happening for fluconazole. So, however, we got like more experiment done for the caffeine that has been because we had like some more data on the adsorption capacity. So this adsorption capacity has been done by one of my colleagues, Ivan Koziatnik, and and the paper is in preparation. But he has been doing the adsorption capacity of wheat straw pellets activated biochar and softwood pellets activated biochar, and we could see that softwood pellets had a lower sorption capacity compared to wheat straw pellets. And we, of course, correlation is not causation, so we don't really know, but we could hypothesize that higher adsorption capacity would mean lower degradation. So as a conclusion, here we can see that the hydrothermal treatment has quite a good potential to decontaminate those spent carbon, and at quite high temperature, so more than 240 degrees. We also have seen that the char is kind of protecting the adsorbed contaminants from the degradation, and the okra act as a catalyst above 240 degrees Celsius. So if you sum it up, I think that it's a good start to find some engineered biochars that could either lower the temperature of degradation, or that could even at the end be used for recycling properties, for the optimized recycling properties. In the end, we still are now currently doing the analysis of the degradation product. And the reason is because when we did the experiments, when we measure like those different contaminants at the end of the experiment, we only targeted those 10 contaminants. We didn't target the product of degradation. And it's quite important to know if we are really creating a worse chemical than the one that are in the water for now. And then finally, we were thinking about doing a regeneration cycle. So Christian is doing that at the moment. And with that, it would be a great way of reusing those shards for a certain amount of time, like maybe 10 cycle maybe, or I don't know. For the moment, we are still working on the data. Thank you for the attention. And as you can see, if you want to visit Umea University, that's the image behind that you usually see every night. Thank you.